My name is Dan Brimberry. I'm a clinical research coordinator in the Cedars-Sinai MAST Program, and I'm presenting a study that foreign travel is associated with changes in the small bowel microbiome and with elevations in anti-vinculin antibody levels. Foreign travel impacts gut microbial populations due to food consumption or exposure to enteric pathogens like pathogenic E. coli or C. jejuni. These enteric pathogens can cause severe travelers' diarrhea and produce cytolethal distending toxin B, CDTB. Antibodies to CDTB cross-react with vinculin, which is associated with post-infectious irritable bowel syndrome. However, there is little additional understanding of the role of foreign travel on the small bowel microbiome. In this study, we explored differences in the small bowel microbiome, as well as anti-vinculin antibody levels in subjects reporting foreign travel compared to those who did not report foreign travel. When comparing the foreign traveler group to the non-traveler group, there were no differences in small intestinal microbial alpha diversity or beta diversity. No differences in age or sex were observed. However, the foreign travel group had significantly lower BMI compared to the non-travel group. For those subjects with available ABS-VCL levels, more than 50% of the foreign travel group tested positive compared to only 25% of the non-travel group who tested positive. After adjusting for BMI, there were 30 significant differences in absolute abundance within the core duodenal species between the foreign travel group and non-travel group. Notable differences in the foreign travel group include higher absolute abundance of Bacteroides species such as B. fragilis, which produces B. fragilis toxin, and B. stercoris. Also identified were increased absolute abundance of Escherichia coli and 15 different species from the genus Streptococcus. 1. Foreign travel is associated with increased prevalence of anti-vinculin antibodies. 2. Foreign travelers have significantly increased absolute abundance of species from genera Bacteroides, Streptococcus, and Escherichia. 3. Future studies will examine how dietary history and location of travel influence the duodenal microbiome and propensity of developing vinculin autoimmunity. Thank you very much for allowing me to present my poster to you. I encourage you to check out the other research from our group at DDW 2026.